back to another video of the pulsating biology. We've already covered this chapter, that is the biological molecules. This video is the basically the crash course for the chapter biological molecule, and we will be covering and revising all of the topics that were included in this. Well, we'll begin with the very basic that the biological molecule include four major types, that are the protein, carbohydrate, lipids, and the fatty acids. So let's begin with the protein first. How the subunit of protein is amino acids. When many amino acids joins together, they give up a macromolecule. That macromolecule is known as the protein. So proteins are basically made up of the monomers of the amino acid that joins together to form a macromolecule, that is the protein. Alright, so how the amino acids are basically the micromolecules of the amino acid are joined together. They are joined together by a peptide bond. This is the bond that is between the amino acids that keep them joined together in a structural format that helps them to develop into a macromolecule. Well, this peptide bond is between the amino acid. There are approximately 20 different amino acids that are formed within the protein. Now let's talk about the amino acid structure. Since I've already told you guys that the amino acid basically joins together to form a protein, let's talk about the structure of amino acid. So amino acid structure contains the amino group, it contains the R group, and it contains the carboxyl group. In the carboxyl group basically mean the COOH group, which is the carboxyl group, and in the R group we may contain any carbon group. Let's talk about it by giving it a review of this structure. This is the central atom that we say is the carbon atom. Then we have the R group, as we have talked about in the amino acid structure. We have the R group. Then we have the amino group. The amino group means the one that consists of one nitrogen atom and two molecules of the hydrogen atom. This gives us the term for the amino group. And in the amino acid structure, we have a constituent for the amino group that says nitrogen, and the two hydrogen molecules that constituents to be upper amino group. Let's come on to the right side of the structure that tells it about the carboxyl group, which contains the two molecules of the oxygen, one hydrogen molecule and one the carbon molecule that makes up the COOH, that stands for the carboxyl group. Well, this is the basic structure of the amino acid that contains three things, that is the amino group, the R group, and the carboxyl group that makes up together an amino acid structure. All right, let's talk about the carbohydrate. The next biological molecule to be talked about is the carbohydrate. What are carbohydrates? Carbohydrates are basically the hydrated carbons. That means that the carbons that are basically constituent of hydrogen and are full with the water content, we say that they are termed as the carbohydrate. So carbohydrate basically are made up of the sugar molecule. When we talk about the sugar molecule, we need to know if they are made up of one sugar molecule, we call it as monosaccharide. If it's made up of a two, then we talk about is it a disaccharide. If it is made up of not more or more than one or two, then we call it as a polysaccharide. As poly means that many, and mono stands for one, and whereas disaccharide means two. So the saccharide term means the glucose, or you can say the sugar molecule, whereas the mono, di, and poly tells us the number of sugar molecules that are included in per carbohydrate. Alright, let's talk about the monosaccharide in detail. So the term monosaccharide means one sugar molecule. In the very common example of the monosaccharide is the glucose. And use the exam purposes, we need to remember the examples of the carbohydrate. So the examples for the monosaccharide is the glucose, whereas the example for the disaccharide includes sucrose, lactose, and the maltose. We do remember the how they are made and what are the constituent of sucrose itself. So sucrose is formed when the glucose and fructose are basically joined together. When glucose and fructose are combined together, it gives us our sucrose. When galactose and glucose joins together, it gives us lactose. And when glucose and glucose molecules join together, which means the monomers of glucose and the monomers of glucose again join together among themselves, they give us a term that is the maltose. This is the sugary content. Let's talk about the polysaccharides. The polysaccharide consists of three examples. That is the starch, that is the glycogen, and that is the cellulose. So the starch is basically a storage molecule that is very commonly found in the plant. Whatever the glycogen, so glycogen is majorly found abundantly in the liver and the muscle cells of a human beings or any organism. When you talk about cellulose, cellulose is a structural molecule that gives the plant cell its shape. That is a very important constituent of the plant cell wall. That cellulose is present there. This is all you need to learn about the carbohydrate and how they are termed as the hydrated carbon, with their three very common examples, that is the monosaccharide, the disaccharide, and the polysaccharide, along with their example. All right, let's learn about the biological molecule that they are the chemical compound that are formed in the living organism. That why they are termed as the biological molecule. The biological molecule basically mean that they are made up of very common constituents that are formed or the elements that are formed in our environment, that is the carbon, hydrogen, and oxygen. But the term in the biological molecule is not similar for all the subgroups. Let's talk about each of them first. Let's talk about the carbohydrate first. So carbohydrate, as I've already told you, that the hydrated carbon, that means they contains carbon, hydrogen, and oxygen. Similar goes with the lipid. When you talk about the lipid also is made up of the carbon, hydrogen, and the oxygen. But when you talk about the protein and the nucleic acid, the same does not apply in these. Well, the protein consists nitrogen and the sulfur as an external element, whereas the nucleic acid consists nitrogen and the phosphorus as an external animal element. So I've already told you guys to learn it the protein, that the protein is made up of a cons, that means the carbon, hydrogen, oxygen, nitrogen, and sulfur. This is the common abbreviation that is commonly used for learning how the protein constituents are made up of. And in the nucleic acid, we say it as a pons, that means phosphorus, oxygen, nitrogen, carbon, and the hydrogen. That means it contains the CHO element, that is the carbon, hydrogen, and oxygen. But along with that, it contains the phosphorus and the nitrogen. And if you talk about the protein, then it contains nitrogen and the sulfur, which have their own several purposes. And this is how you need to learn about the chemical compounds or the chemical elements that is found in the four subgroups of the biological molecule. All right, let's jump on to the third very common group, that is the fat or the lipids. There's not much to learn about the fats or the lipids. They are also made up of the carbon, hydrogen, and the oxygen. There does not have any other further element along with that. They are the biological molecule that are made up of carbon, hydrogen, and the oxygen. They're very simple to learn. The very common example that we often have heard about is the testosterone. This is found in the animal and the plant cells both, and the formula that you need to learn for your exams is the C57H10 and O oxygen. This formula tells us that there are 57 carbon molecules or the carbon atoms, there are 10 hydrogen atoms, and there are 6 oxygen atoms that helps it to be the constituent of the fats. Now that helps it to be the constituent of the testosterone itself. So this is a very common example, and there are several MCQs linked with this. Do remember this, please. Let's talk about the sources of fats. Where we can get the fat or the lipids. Let's give it a view that the very common food constituent that gives us the fat or the lipids are the fatty, mostly consist of the fatty content. Then we have some healthy peanuts and the beets, that is the butter, cheese, and the fatty meat. Then it comes the olive, nuts, and the peas and the beans. Now, these are the very common intakes in our daily life, which are the very rich source of the fats or the lipids that gives our body its requirement and fulfill the requirement of the balanced diet that includes the fats and the lipids. When it comes to the fats and the lipids, the basically fats and the lipids are made up of the fatty acids and the glycerol. These are the monomers of the fats and the lipids. The fats are the made up of the fatty acids, while the glycerol is the monomers of the lipids. The lipids are mostly found in animal and the plant cell both, and they are commonly made up of the carbon, hydrogen, and the oxygen. This is the beef fat that we have already discussed in the last slide. How 57 carbon atom, 110 of hydrogen atom, and 6 oxygen atom gives us a very common fat content, that is the testosterone, which is present in both animal and the plant cell. This is all you need to learn about the fats and the lipids. All right, let's come on to the functions of the fats. So, fats is the basic source of energy. It fulfills the requirement of the or the proportion of the fat or the lipid that is required by our body and gives us the full source of energy. When we come on to the fat being an insulating material, so basically, fat forms up a layer on our organs, on our cells to protect them from the external environment and prevents the heat loss. So, it is termed as an insulating material along with the source of energy. The third pointer says that it is a solvent for the fat-soluble vitamins. The fat-soluble vitamins does not dissolve in a very common soluble content or in the water. It only dissolves. It is only able to be dissolved in the fats and the lipids. So it is a solvent for all the fat-soluble vitamins in order to perform many metabolic reactions. So the fourth pointer says that it is an essential part of the protoplasm. We already know when it comes to the protoplasm is a common content that is found in the cell wall, and in the cell wall, the fat contains a very important role as it prevents the heat loss from the cell. So these are the four pointers that tells us about the functions of the fat. All right. So the fourth subgroup that we had is the nucleotide. So the parts of the nucleotide includes phosphate group, it includes the base and it includes the sugar. When you talk about the base and the sugar, in the sugar we say that there is a ribose sugar which consists of 5 carbon atoms. There is the ribose sugar. When we talk about the phosphate group, we have the phosphate group and we have the base. In the base, we have four kinds of the base, that is the adenine, thymine, cytosine, and the guanine. Adenine and thymine are mostly linked together, whereas cytosine and guanine are mostly linked together with the several bonds that we're going to talk about. That adenine and thymine consist of a double bond, whereas cytosine and guanine consist of a triple bond between them. But before talking about that, let's talk about a very common subgroup of the nucleotide, that is the DNA and the RNA. Well, the DNA stands for deoxyribonucleic acid that consists of the genetic material, whereas if you talk about the RNA, this is a ribonucleic acid. What is the difference between the DNA and the RNA? So, DNA is a double helix structure. This is the double helix structure. When you know about it, this is the single helix, this is the double helix. When the two helix cross each other, we call it as a double helix structure. So this is the double helix structure that consists sugar, phosphate, and the bases. And between them is the bases that we say nitrogen, adenine, and the thymine, so cytosine, and the guanine, along with their amount of hydrogen bonds between them, and they are bind together with a very common bond, that is the hydrogen bond. Please do remember this. Like I've already told you guys that the about the double and the triple bond in the adenine and the thymine, we have a double bond, whereas cytosine and guanine we have a triple bond. Similarly, in thymine and adenine we have a double bond, whereas in cytosine and guanine we have a triple bond. This is how they are bounded together with the help of a hydrogen bond that helps them keep together in a single space. And this is all you need to learn about the nucleotide. Since we have discussed about most of the subgroups of the biological molecule, let's come on to the test of the biological molecule. Number one test is the Benedict's test. Let's talk about the Benedict's test. So the Benedict's test is basically the test for the reducing sugar. And how is it is performed? We take the 2 centimeter glucose solution, and in the similar quantity, we need to take the Benedict solution. Whenever we mix together, we need to heat it up at 80 to 100 degrees centigrade. When we heat it up and boils it, so basically it gives a change of a color. Whenever there is a change of a color, please do note this. Whenever there is a change of a color, we say that the test is basically the test is successful. And when it does not change the color, we say that the test is not that much successful. So the first color was in blue color, since the Benedict solution is blue colored. When the color changes from the blue to brick red, we say that there is the sugar present. If the blue does not change from the blue, we say that there is an absence of a sugar molecule. But the Benedict test goes through the several stages. It goes through the four different stages, which just gives us the four different colors. Let's say about when it is turned blue, we say that there is a no change that takes place. When we talk about the green, there is a tracing of the reducing sugar, that it might turn into a great amount of the reducing sugar itself. When we talk about the orange, we say that there is a moderate quantity of the reducing sugar. But when it turns to brick red, we say that there is a large amount of sugar present, and and the test is successful. This is all we need to learn about the Benedict's test. Let's talk about another test. So the next test is the ethanol emulsion test. This is the second test for the biological molecule which is related to the lipids and the fats and its dissolution. So what happens is that we take any food sample, whatever the food you are eating, whether it is a biscuit, whether it is a chips, whether it is any store item, whatever you take, whatever we do with that is that we add ethanol and then we add some amount of the water. Do remember this, there's the ethanol and the amount of water is similar in the quantity. So what happens is that it forms up a clear structure or the clear solution. We say that there is no quantity of lipid and fats present. But if it turns into an emulsified solution, which means that it turns into a white cloudy solution, then we say that there is a presence of lipid. White cloudy solution that is termed as the emulsified, then we say there is a presence of the lipid. This is a very simple test of the lipids and the fats. The third test we have is for the protein, that is the Burette test. In this test also we take a food sample and then we crush that sample with whatever you are eating. Just take that sample and then we crush that sample. We add sodium hydroxide in it and then we add the copper sulfate. The copper sulfate is majorly known for being very blue in color. So that means that the our starting quantity would have a blue color, and if there's a change of a color, then we say that the test is successful. So the first color is the blue in appearance is the blue, and if it does not change, then we say there is a no amount of protein. But if it changes to purple, then we say the protein is present. Very common example and the very basic thing you can learn it from is that P for purple, P for protein, and P for present. This PPP tells us that there is a change in the color, that is the purple, and is the presence of protein. Also, the color intensity tells us that the amount of a protein. If the purple is very dark, then we say that there is much amount of a protein present. But if the purple is not that dark, or it's a lilac color, then we say that the protein is not that present. All right, the last test that we're going to discuss is the iodine test, which is for the starch. All right, so in the similar manner, we're going to take a food sample and then we're going to crush it. Once we have crushed it, we are going to add an iodine solution in it. The iodine is brown in color. If it does not change the color, there is no presence of the starch. But if it changes its color to blue black, we say that the starch is present. This is a very easy test. We have four tests in total. We have iodine test, we have emulsion test, we have Burette test, and we have Benedict test. We have discussed each of these tests and their change in the color. I hope that you have found this video very helpful and I have revised all of the topics in single video. That's it for this video. Thank you very much.